Well, welcome back, everybody, to Saddlebrook Science Cafe Series, Fall 2018. This year, our theme is Mysteries Revealed, Discoveries in Chemistry. I'm Nicole Leitner. I'm a PhD student at the University of Arizona. I study ecology and evolutionary biology, and I will be here to announce tonight's speaker. I'm really excited to be here tonight with you guys, especially because recently I have been furiously writing my dissertation so that I can graduate in December, so I haven't seen the light of day in a while. It's nice to get out of the house. Thank you. Anyway, I'd like to thank the DesertView Performing Arts Center staff for setting us up here so beautifully, and thank you all for coming tonight. The Science Cafe is brought to you by the College of Science at the University of Arizona. It's one of 5 venues, and each one has a different theme, and they are all completely free to the public. Please take one of the Science Cafe brochures on the table out there to get more information on some of the upcoming lectures. And please feel free to take as many of the other brochures of our various College of Science outreach facilities as well. We have plenty more where they came from, and it'll give you a chance to see some of the stuff we have to do around town. And also on that table, those of you who have been here before are probably familiar with it, but we have these evaluations that not only give you the opportunity to give us feedback, but also allow you to provide us with your email address so that we can send you College of Science updates on upcoming lectures and whatnot. So tonight's format is the usual 45-minute presentation, followed by an audience Q&A where Darby and I will walk around with the microphones. And then Roger can let you know if he's available after that for one-on-one questions. I'll let you feel that. And with that, I'd like to introduce our speaker tonight, Roger Miesfeld. His talk tonight is Everyday Biochemistry, How It Works and Why It Matters. And Roger is a distinguished professor and department head in the Department of Chemistry and Biochemistry. He grew up in San Diego and earned his bachelor's and master's degrees in cell biology from San Diego State University, a PhD in biochemistry from Stony Brook University on Long Island, and was a postdoctoral fellow at University of California in San Francisco, studying mechanisms of steroid hormone signaling. And he's interested in how cells communicate with each other using chemical processes mediated by proteins. And Roger has always actually been interested in biomedical science, having come from a medical family. Father and grandfather are both physicians. And he came to the University of Arizona in 1987. I was 4 years old. To work on childhood leukemia in the Arizona Cancer Center, and later worked on asthma in the Asthma Research Center, and currently works in the area of tropical medicine, studying how mosquitoes make desiccation-resistant eggshells with the hope that we can disrupt this process and decrease mosquito populations in areas of the world where mosquito-borne diseases are impacting health. In his free time, which might surprise you that he has any, he has become quite good at home brewing beer and hard cider. His son, who has recently finished his Master of Architecture degree at the University of Arizona, lives in Tucson. He got him into the whole brewing business. And Roger has kept at it, even though his son is now too busy to help out. And Roger is even adding an outdoor kitchen to the house with a room for the brewing equipment, so it's going well. Tonight, Dr. Miesfeld will discuss how biochemistry is the chemical science of life on Earth. How it grew out of the desire of chemists to unravel the mysteries of life. What they discovered at the end of the 19th century was that life can be explained by the same physical laws that applied to the geological crust of the Earth, and it did not require an ethereal or spiritual explanation, as was the thought of the day. Tonight, he will give you examples of everyday experiences we have in which we know how biochemistry works and why it matters to us. And with that, I'd like to introduce Roger Miesfeld.
Thank you very much. Well, welcome. I guess we're on here. You can hear me. Thank you very much for coming. I'm very happy to give the inaugural lecture tonight for the series for this academic year. The Department of Chemistry and Biochemistry was invited to participate, and being the department head, I quickly rounded up some of our best colleagues to ask them to come up. And so over the next 6 months, you'll have the privilege of getting to know some of our faculty. So I actually am as Nicole read off the bio that I gave her, I'm a biochemist. I've worked in medical sciences my whole career. Tonight, I'm not going to talk about mosquitoes. I was tempted to throw a few really cool mosquito slides in there, but we're going to stick to the topic of introducing the chemical science of life, and in particular biochemistry. In the last 5 or 10 years or so, I actually got involved in textbook writing. I've actually written 3 textbooks. And my latest textbook, which is published by Norton Publishing in New York City, is a typical big textbook. Most of you who have gone to college, and back in the day when I went to college, as many of you did, we'd have these 3-pound textbooks. This actually is, I think, 4 pounds. Everything is an ebook now. My students all have ebooks, and they use the ebooks to search. We occasionally still buy printed books. So what my talk tonight is going to be the theme of the book, which is biochemistry, how it works and why it matters. Why is that the theme of my book? Because over 30 years of teaching biochemistry, I found that students are more interested and more probably capable of understanding biochemistry if they have a reason to understand it. And so I'm going to give you some examples tonight. You're going to see we have some props up here, just in case you can't see in the back. A loaf of bread. This is from Beyond Bread, which of course is an artisan bakery here in town. These are going to be gifts, and Nicole is going to help whoever is brave enough to answer my questions. This is a bottle of one of Miesfeld Brewery. We're actually called Bobcat Dog Brewery in Tanque Verde. My son and I got going, and my daughter bought us labels, so we have 20 labels to put on beer. This is actually a hard apple cider that I made, bottled it last week. It's about 4 weeks old. My oldest daughter is getting married in 3 weeks, and so we made some hard cider for her wedding, which will be at Hacienda del Sol. And then this is the Beano, which is some of you might know about. We're going to talk about what this is and where it comes from, and that'll be an example of everyday biochemistry. And then last of all, I have a bag of what's called Sour Jack's candy, and it's a super, super sour candy, and we're going to talk about what we know about, how they made this candy and what we can learn from that in terms of biochemistry. So that's kind of the overview. And you have most of you got a card as you walked in. A lot of you folded them already correctly, so you're way ahead of the game here. I will show you how to fold it if you haven't folded it. And we're going to have fun tonight. We're going to do active learning. So kind of the 2 things I hope to communicate to you tonight. What is biochemistry? Why is it so much fun? We're going to have a ton of fun. And then the second thing is how I teach my class. So I teach, my class is about 2 or 3 times larger than what's in here tonight. My classes normally would be all the seats are filled. So today I taught a class with 310 students. We were in the Gallagher movie theater down at the Student Union at U of A if you've been there. And I've also taught 500 students in the environmental science building. So why so many students? Biochemistry is a foundational science. Medical schools now require 1 semester, absolutely required, of biochemistry to even put in an application for medical school. Great for my textbook. We're selling tons of books, and my classes are full. So really what got me excited here was, a lot of people dread biochemistry, dread organic chemistry. Anybody ever take biochemistry? Okay, good. See, you guys are going to have fun. This is going to be cool. All right, so enough of that. Let's go ahead and get started. The Department of Chem Biochem, I'm going to give you a little bit of an overview because when the other 5 professors come, you'll have a little bit of a sense of kind of what we are. This is a picture that was taken, I think it was August 20th, actually. We actually have almost 1,000 undergrad students have majors in chem or biochem. This is just a small group. We call them ambassadors. They're our most enthusiastic. And I think we had an ice cream social that day. So it was just kind of a great picture, a very recent picture. So some of you picked up, I noticed, the brag sheet that we have out there that we use for public relations and alumni giving. This kind of just gives you a quick overview of what we are. We came up with this sort of a motto. Our research portfolio stretches from bugs work on mosquitoes, to batteries. We have somebody making new lithium sulfur batteries. And from cancer cures, we have organic chemists that are making compounds. To catalytic chemistry, we have faculty, and one of them, Tomas Giannetti from France, is going to come and give a talk. And Tomas is studying reactions with catalysis that are 100 times better than what we have today. Then on the right-hand side, just to give you the scope of kind of what it means to be department head of chemistry and biochemistry, 53 tenure track and teaching faculty, 165 PhD students, 65 biochem, 325 chem majors, 50,000 student credit hours. A credit hour, a 3-unit course is 3 credit hours. So if you take 100 students, right, you're going to have 300 credit hours. So we teach 50,000 student credit hours a year, mostly Gen Chem and Organic Chem. My biochem class alone is 1,500 students a year, so that's 4,500 student credit hours. And then lastly, how big of an operation is this? About $20 million a year. About half is the money that we get from tuition and state appropriations, and half is grants and contracts. All right, so tonight I'm just going to focus on this. Part of it comes from, believe it or not, if anybody was in business, if you were in science business or in a tech business, this is partly from a talk that I gave in Portland, Oregon a year and a half ago. The company Norton Publishing is a large independently owned publisher. It's my opinion, one of the best out there. And they invited me to their summer sales meeting. And what they told me, they said, your book is beautiful, looks great, we love it. But if the travelers, which is the name they give to their salespeople, if they don't get excited about your book, they're the boots on the ground. If they won't go into a biochem professor's office at University of California, San Diego, then they're not going to sell your book. So I had an audience of salespeople, basically, and I gave a sales pitch on how and why they should sell my book. So I took some of the slides from that. So there's a little bit of sort of background on the book that you'll see. The how it works and why it matters. For those of you, quite a few, you raised your hand, took a biochem class. So in the old days, when I took biochem, it was memorize the glycolysis, memorize the citrate cycle, what is acid-base catalysis, that kind of thing. Even I'm bored by that. And so what I wanted to do was, coming, like I said, from a medical family and always being interested in medical research, I know it's exciting and I know it matters. So the question was how to connect to students. So oftentimes, it's not so much just a drug, to cure a disease. It's actually stuff in their everyday lives. Okay, so that's the theme. This everyday biochemistry was a word that I coined. Actually, I was giving a lecture in Social Science 100, which is one of our largest lecture halls at University of Arizona. And my editor at the time was trying to get me to how are we going to package this thing. And I kind of was, at the time, which was about 5 or 6 years ago, every day anything was sort of happening. So in my head, I remember one day I gave a lecture and I go, hey guys, I think we need to really think about, for example, we were talking about Beano, and I said, this is an example of everyday biochemistry. So we sort of went with that. I have worked with a group in New York to try to figure out how to make a lecture like this or a presentation like this work for students so that it doesn't just become sort of a stand-up show. And so what we do in the classroom, and we're going to do it tonight, is we do what's called active learning. Active learning is where the students participate. Doesn't matter if it's a room of 50 students or 500, they participate. And the way that I do it is I take students who have taken my course the semester before or the year before. They got an A in the class. They're still on campus. They're juniors or seniors. I invite them to be what are called preceptors. Preceptors are undergrad students who know the topic, and they become part of my team. So if this were my classroom, I'd have 12 preceptors or 15. They would be sitting in different parts of the room. And when we go to do the active learning with the cards, which I'll do in a minute, the preceptors would stand up. We'll stand up, they do stand up, and they go around and they help the students understand. So that's the way to break the course down. And that's what that video is on the left-hand side. It comes out of a teaching company, and the title of that video had to do with how to make active learning work in a large classroom. Okay, so what was my journey? I forgot that I gave Nicole all this earlier in my bio, but so the question was, and I'm confessing to you that was the slide for the sales pitch. Why did I even write the book? I grew up in San Diego. I was a surfer. I wish I still was a surfer. I love the water, but it was a great time to be in San Diego. I went to San Diego State. My father, who was a physician, couldn't understand why I didn't go to UC San Diego. Because that's the premier school. Why aren't you going there? And I said, well, I don't know what I want to do. I don't want to go to a school where I'm going to fail. I'll just go to San Diego State for now and I'll figure it out. And what happened was I got into a lab early on, working actually on tomato root tips. We were trying to study aging of why a root stops growing after a while. You can cut it off and it'll grow and it'll stop. So I got excited about research, and then because of that great experience at San Diego State, my mentor encouraged me to go back east. So I went to Stony Brook and did well, and had a good run there. And then I went to UC San Francisco. So as I went through my training, I always thought of myself as a teacher, as a professor. And so I ended up doing that, and of course, do lots of research. I've been at U of A since '87. So we heard Nicole was 4 years old. I was only 6 years old, so that was pretty good. Made it through department head. I showed you how big of a job that is. It's a service, I guess. I'm giving back. And I recently was appointed a university distinguished professor. I've co-authored 3 textbooks. The last one is the biggest, and although it took me 15 years, it became a hobby. Every weekend I would work on the book, and my wife said that's a really weird hobby to have. So, but I got it done. So we'll talk about that. And so the little wiggle over there, you're going to see a little wiggle later on. I had a wiggle there because it turns out my wife Elizabeth is sort of a graphic artist, and she did all the graphics for this textbook, which was pretty cool. That was published by Wiley and Sons, and Wiley and Sons is sort of a big conglomerate, and they kind of made us do our own figures, whereas Norton, which is a terrific publishing company, W.W. Norton, they did all the figures for us, so the figures are beautiful. All right, so what is the discipline? So part of my job tonight is to sort of orient you to what the future talks are going to be. You're going to hear some really interesting talks on pain receptors. You're going to hear some talks on drug discovery. And so the first question then is, what is biochemistry? And so I'm sort of leaving chemistry to the side. I'm just going to focus on biochemistry. So biochemistry aims, it's our goal, it's our aspiration, to explain biological processes at the molecular and often the cellular level. So we work inside a cell. As the name implies, biochemistry is at the interface of biology and chemistry. Could be a chemical compound. This is citrulline. It's part of the urea cycle. We think about cells. This is a macrophage ingesting bacteria. It's the way our body is going to fight off disease. And then on the right-hand side there is one of the pictures from the book. I was explaining in one of the chapters in the book, which you can buy on Amazon.com, by the way, is how humans have more signaling genes. We have more genes in our genome than a yeast cell does, than does a flatworm. So as organisms get more complex, a lot of the complexity is actually cell signaling genes, how cells communicate. So that's an example of kind of how I tried to visualize that. It's a hands-on experimental science. I spent all my time in the lab, and when I first opened my lab here at the Cancer Center, for the first 5 years, I was in the lab working side by side with my grad students. This is a sort of kind of saying the way we get proteins nowadays is we grow them up in bacteria, or we isolate them from human cancer cells. When I started biochemistry back in the old days, and there's people in here may have done it even before me, you would go to the slaughterhouse or go to a butcher, and you would get a big calf liver, or you would get a heart muscle, and you would grind up that animal tissue and isolate the protein. About the late 70s, early 80s, we had the DNA revolution. Genes were cloned, and those genes could now be put into bacteria. The bacteria would crank out the protein. My work at UC San Francisco in the mid-'80s was I cloned the gene that encodes the cortisol receptor. So if you've ever had prednisone or triamcinolone acetonide or dexamethasone or cortisone, the gene that I was lucky enough to, well, lucky, I worked hard. I picked a clone off a Petri plate, a bacterial clone, and I picked the colony off the plate through an experiment that I did. It actually contained the coding sequence for the receptor that the steroid binds to. And so I went on and came here to Arizona, but I spent a lot of time just doing it. So a biochemist works in the lab. You're going to hear a lot of that from my colleagues. We certainly are bioinformatics people. We spend a lot of time at computers. We have faculty that, of course, write their own algorithms. We can't just anymore just stay in the lab. We need to analyze it. So where do we see biochemistry? Well, I call it an applied science. I've always been interested in medical science, but I also hold a patent. We have another patent pending. These are compounds that interfere with mosquitoes. And so if you look across the board here, Nicole here kind of works on environmental science. Biotechnology are sort of those Ventana Medical Systems here in Tucson, high throughput genomics. Another company here, we used to have Sanofi that was up the road in Oracle, down in Oro Valley. Agriculture, how do you make a tomato that doesn't over ripen? Pharmaceuticals, of course, an easy one. Clinical diagnostics, how do you diagnose a disease early enough that you can actually interface with treatment? Those are all discovered in biochemistry. And the last one is a commercial product. That one is actually one that we'll talk about in a minute. Okay, so one of the things I can't really show you, I was going to try to do a demo for you, but it wasn't quite working. I didn't want to mess it up. But the proteins, which a lot of biochemists work on proteins. I mean, I do work on steroid hormones, I do work on lipids, but proteins are sort of the bread and butter of a biochemist. A molecular biologist or a geneticist is going to work on DNA, but biochemists tend to work on proteins. And the way a protein is analyzed or characterized is really kind of two ways. One way is to purify the protein to homogeneity and purify it and put it at a very high concentration, and it'll actually crystallize out. It'll form crystals. You can take the crystal's small, millimeter, and all the protein molecules are lined up in a lattice. You shoot an X-ray at it, you turn the crystal, shoot another bunch of X-rays at it, and you collect on the backside the film that has where the density of electrons are. The computer can reconvolute that, it's called Fourier transformation, and turn it into a three-dimensional structure of where the atoms are. We then take those computer programs and we can now visualize using visualization tools where the cancer drug might bind to a protein and then try to design a better drug. So a lot of my textbook, a lot of what I teach in my class is the three-dimensional structure of proteins. We use models. Some of you may have seen some of these before. There's a ribbon structure. Proteins aren't actually ribbons. This was invented by a computer scientist using sort of ribbons as almost a hieroglyphic, but exactly where the ribbon is and exactly where that DNA strand is, that's exactly where it is in the crystal. So it's not drawn as a cartoon, it's actually there. And if you look over on the left-hand side, that shows you really what the electron cloud that's around the atom. So there's kind of two ways to look at it. And then a lot of the book, because of my background and certainly a lot of the students that I teach, and one of the reasons the book is selling so well is that a lot of the professional health students and interest has to do with human health. And so this is an example that's in the textbook. It has to do with sickle cell anemia. We use the structures that we learned from hemoglobin and sickle cell globin to understand why a person who has sickle cell anemia has this anemic phenotype. And what you can see over here, got my pointer, this is a sickled red blood cell. This is actually from a scanning electron micrograph. So again, it's not a cartoon. Excuse me, that red blood cell is sickled because the fiber was formed between the globin protein. The globin protein is not supposed to form fibers, but when it has a mutation, this is a mutation down here, it forms a hydrophobic patch, and now two adjacent molecules come together. And I'm going to suck on a Ricola here. which I do in class quite a bit. The students sometimes yell out, "Suck on a Ricola, your voice is cracking." So, all right, I'll do the same thing. All right, so there's an example of the molecular interaction. The mutation is right there. It's a valine. It's not supposed to be a valine. It forms these polymers. They form these long polymeric strands. They break the cell. The cell breaks. It's a sickle cell. Okay. All right, so let's start the fun part. All right, so biochemistry is a fun course to teach. You can tell that I'm having fun, I hope. But I say it's only fun if done right. And I would venture to say my biochem course I took at San Diego State in 1976 was pretty boring, although I sort of was intrigued by it. I didn't really appreciate having to memorize a bunch of pathways. I didn't really learn too much. So I say it's fun to teach if it's done right. And so the question is how to do it right. What do I mean by that? Okay. So these are my students actually at the University of Arizona. This was actually in that some of the brochures that went out there. You guys are going to do this. The deal is that you have it folded, and the way this works is when I ask a question, you're going to pick your answer and you're going to hold it to your chest. You don't want your neighbor to see it. Okay, because it's your private answer, but I can see it perfectly. So I'll be able to look out, and if I know what the answer is, and I see a bunch of answers that are correct, I feel like you guys are on track. If I see all 4 different answers, or I see a bunch of answers that are wrong, then we could stop and kind of go from there. When you get a right answer, and Nicole gives you the microphone, if you can explain it, you actually get to have one of the props. I always like to show this one because this guy wasn't quite sure what he was doing. Notice his letter is upside down. I get to pick on him actually. Okay, and then this is a preceptor. This is Natalie. She actually went on to medical school, and so she is helping the students in my class during one of the active learning sessions. So that's how it works. Wait, why is everyone smiling? Is biochemistry really that much fun? Of course it is, right? But you'll be the judge of that. Okay, so we're ready to do this. All right, so first of all, I'll show you in a minute. But basically, I made it. I think a lot of people were doing this. I worked on this the other day. When I first did this, and that's why the student had it upside down. Actually, I had A, B, C, D, and then the other day I was thinking before I come down up to Saddlebrook, I'm going to try to help you out here. So if you fold it this way first, so it says fold first. So now I have B, C, and D, A. And then I just fold it the second time this way. This color didn't print very well, but A, B, C, D. And so what you're going to do is you'll look at the multiple choice question. You'll pick your answer. So I'll say the answer is A, and I'll say, okay, show me your answer from there. So that's where we're headed. All right, so we're going to do 3 questions. I think I have enough time to do this. We'll get through it, and you'll have so much fun. You're not going to want to leave anything out. 3 questions, 3 props. Plenty of prize. First one, what do a loaf of artisan bread and a bottle of homebrew cider have in common? And I'll give you answers, and then we'll have a follow-up question, which is typical. How are they different? Okay. Second question, how many lemons does it take to produce a bag of Sour Jack's candy? All right, we'll have a follow-up question for that. And then the third one is going to be, why does broccoli cause intestinal gas? So why? And then the follow-up question, how does Beano work? Why does that help you alleviate that? Okay, so that's what we're going to do. All right, so first one, fold it. I did this little thing, fold in half. All right, then you fold it again, and then you should have your card. All right, so I think everybody's on track there. All right, 1 of 4 possible multiple choice answers. All right, so now, Nicole, I'm going to try not to give away the answer. Now, one of my students always laugh because, Dr. Musel, you went to the next slide. You should have waited. So I have a cue. My cue is it says think, pair, share. Anybody was a teacher K through 12? Okay. Anybody was a teacher higher education? Okay. All right. So think, pair, share was one of these things that got coined. But it's things we did when we went to school. It basically says you could talk to your neighbor, you get the question, you think, and then you pair. You say, hey, Johnny, hey, Joni, what did you think? And then you share. Okay, so you don't have to pair, you have to think and you have to share. So pairing is completely up to you. All right, so when you see the Thinkpairshare, when it wiggles, that means you get a couple minutes to try to figure it out, okay? All right, here we go. What do a loaf of artisan bread and a bottle of homebrew cider have in common? Here are your choices. Okay, so you're reading those. I'm not going to read them to you. And then when I say it's time, you're going to do what these guys did. So we'll do Thinkpairshare and then Thinkpairshare. So go ahead, talk amongst yourself. You get to pick A, B, C, or D. I want you to think about it a little bit, and then I'm going to ask who wants to share outwardly what they think it is, and then Nicole is going to carry this great. This is actually the one. This is artisan bread from Beyond Bread. This one's I think it's I can't remember. But it's the most grainy one. So if you like it, all right. So some people are holding this up already. So and when I saw how many people took biochemistry and 50, you raised your hand. It's pretty easy. All right. So well, I'll just read them off so we can kind of get warmed up here. They both consist of high-calorie fats. They both contain alcohol and water. They both require ethereal spirits. They both are made with fungi. Put your heart thing up there. All right, I'm looking around. Nicole, you're looking around. All right, who wants to explain about D, why you picked D? All right, we got one back there. You picked D, so tell us, explain a little bit more about what you know about how bread and beer.
They're both made with yeast.
Okay, yeast, all right, and I said fungus, but it's yeast, so very good. Okay, do you know what the yeast do?
Well, in the bread, it makes the bread rise. In the beer, it ferments it.
Okay, very good. All right, good. All right, so you got the bread. Congratulations. Did a good. Give her a hand. Did a good job. Okay. The name of the yeast is Saccharomyces cerevisiae. It came out of Berlin. It was the one that Edward Buchner used to show that he could actually crack open a yeast cell and he could still get things to work. So that's how he knew it didn't have to be an ethereal spirit. And then the other two doesn't do with fat, nothing to do with water. Okay, very good. All right, so now we'll go to the next one. How are they different? So we had that they both use Saccharomyces, they both use yeast. How are they different? Here are your choices. And time to go to work. Okay, think about it. You can share. That's Thinkpairshare. See, there you go, more normal. They get progressively harder. The first one a little bit easier. You probably laughed at D. Guys, all right, I see people folding. Folding's good. I see, all right. On the count of three, put it up to your chest. 1, 2, 3. I see C, I see B, I see D. D, we know that, and I see an A over there. Okay, so we got a little bit of everything. All right, so who wants to tell me why they picked the answer that they picked? We don't know if it's right or wrong. Let's just see. Who wants to raise their hand and say why you picked whatever letter you picked? Okay.
I took C.
All right, why'd you pick C?
Because it's fermentation and carbon dioxide is produced by the yeast, and that the bread traps it, which is why it rises.
Okay.
And cider also makes CO2, which is the bubbles.
Okay, it does, as long as you keep a cap on it.
Absolutely.
But
it could be lost, and what's left behind is the alcohol.
Perfect. All right, so guess what? Give him a hand. He wins the bottle. All right, so what's your name?
Eric.
Eric. All right, so I bottled it a week ago, and what happens is when you open it up, and I'm actually going to show you a little bit of a movie. So after it ferments, it's in a big bucket, and so the CO2 is not trapped anymore because I had to open it actually. And then I siphon it into another carboy, and then I put I measure exactly how much. I put sugar in the bottom of the bottle. Some yeast do carry over, and then I cap it. Then I put it in the closet for 3 or 4 weeks. The little bit of yeast that got carried over, they will ferment the sugar that's in there, and they'll recarbonate. So it carbonates a couple of times, but every time you open it to transfer it, of course, it comes out. So when you drink it, I had some last night. It was a little bit like wine because it hadn't gone long enough, but in a week or two, it'll be more carbonated. All right, very good. Okay, so that's the answer. Why don't you get drunk when you eat bread? You don't get drunk when you eat bread. Why don't you get drunk when you eat bread? You know why? Why? The alcohol evaporates in the oven, just pours out. It's just very volatile. It's not going to be trapped in there. And actually, and I just explained to him that, of course, wine is not carbonated, but wine has alcohol. So it depends on how you make the spirit, whether it's going to be carbonated or not. All right, so you have the ethanol in the fermented, and you have the CO2. Very good. You guys are good. All right. So next one, what reaction converts? So then I'm going to teach you biochemistry. All right. So I tell the students, tell me how C6H12OH, which actually that should be O6. Sorry about the H in there. Goes to CO2 and C2H5OH. Glucose going to CO2 and ethanol. Although I did have a chemical problem there. And so this is now what we're going to learn. Okay, so you go to chapter 1 in the book, and we'll go through this. There's glucose, C6H12O6, got 6 oxygens. There are my yeast cells that you told me about, Saccharomyces cerevisiae. That's a scanning electron micrograph taken. It was colorized. And then it goes to ethanol, and you can say the CH2, the methyl group CH3. There's going to be 2 ethanols because we had 6 carbons to start with, right? We gave up a CO2. That's the one on the end there. It either got trapped in the bottle or it didn't. So now I have 2 carbons, plus I have 2 carbons in my ethanol. I had 2 of those. So I added up, I had my 6 carbons. And the question then is, how is the yeast doing that? Okay. And how does that relate to bread? All right. So it's the same in that the yeast are doing the same thing, except as he told me, the alcohol is volatile and you lose it in the oven as it's cooking. But because you kneaded the bread and you got the fibers to trap so that the gas doesn't escape, that's why you get the big bubble, because you spent time lining up those gluten particles to get the bread to form a mesh. And so as the yeast were fermenting the sugar that's in there, then it forms a pocket, and that makes the bread rise. And of course, you need sugar for that to work. Okay, so here's the reaction. Pyruvate is the last product, and I know some of you said you took that class. Glucose goes to 2-pyruvate, and then if the yeast is under anaerobic conditions, okay, not where it can convert everything to water, if you make it under anaerobic conditions, so in the bread, it's trapped. In my brewing bucket, it's trapped because I don't let air in there. And so the enzyme pyruvate decarboxylase, which is an enzyme found in yeast, it's activated. It converts pyruvate to acetaldehyde. And then in the second step, alcohol dehydrogenase, the second enzyme, which is found in yeast, it converts acetaldehyde to ethanol in the presence of the coenzyme NAD. That's a redox reaction. It transfers 2 electrons from acetaldehyde to it's actually an oxidation reaction. It strips 2 electrons off, and you can see that carbon in the middle is more reduced as a result of the 2 electrons coming from NADH. Okay, so there you go. You just learned something. Okay, all right, cool. And you make beer, you make cider, you make wine. All right. Guess what? I told you I do this at home. All right, so on the left-hand side is my front patio, and we live in Tanque Verde Valley. And the question is, what's the name of our little home brew? Bobcat Dog Brewery. And my daughter, who's in advertising and marketing in San Francisco, she wanted to get me my own label. So it says craft beer, crafty science. So she bought me 20 labels. I put them on my beer, and you got one of my precious labels. Why Bobcat Dog? Well, we live at the base of Mount Levin. We live 3 miles up Catalina Highway, right before Snyder, if you've been out there, which is the other end of the world for you guys. There is a turnout. The very first turnout is called Bobcat Dog, which means Frog Mountain. And my kids, they could see that turnout from our front yard. You can just kind of look up there. They went to Tanque Verde schools. And one of them said they couldn't say Bobcat Dog, so they said Bobcat Dog. So we call it Bobcat Dog. All right, so I'm pulling out. I'm over here. What I have it's basically a sock. It's actually a brewing knit. And what I did was I was adding flavor. The hops were in the bag, and it boiled on my front patio there. We had a Bunsen burner, and I was adding flavor. So that's what I was doing. Okay? All right, do I really brew? I hope this works. Let's see. This is the cider that he has the bottle of. So what am I doing? This is after the first fermentation. I'm now siphoning below that yeast. That whole top part is yeast. They've been in there for a week, and I'm siphoning right below the yeast into the second fermenter. So this is a glass carboy. The reason I went into glass is because now this will go 2 more weeks, and then I'll siphon again into the bottle. And I need to be glass so I don't pull too much yeast up. So that's what that is. So yes, I do it. This is in the glass carboy. I try to keep it, it shows 69 degrees. I have a little, I'm like most of the people in here, any hobby that I can buy a toy, I did. So that was the laser thermal thing. My wife, you know, my Amazon Prime account, just keep hitting that. And then I'm in Tucson, so I need to keep it at 65 if I can. Otherwise, the yeast start to make other compounds, and some of those compounds give a bad flavor. And so I try to keep it low, and I don't have it in a refrigerated unit, but I will in my new farm kitchen. I use a fan, and I use the towel, and I get some evaporative cooling. I can't brew in the middle of summer because I can't get it cool enough. We keep the house with our air conditioner, you know, 77, 76, and it really needs to be 10 degrees cooler. And then, of course, I do bottle it and cap it, and he has one of those. Okay, let's move on. All right, second one. All right, so before I forget, I'm going to give this to Nicole so you don't. And I would say that's pure sugar candy. So give it to somebody that you want or your grandchildren or something. All right, how many lemons does it take to produce a bag of Sour Jack's candy? Super sour. Okay, and you're thinking, you know, that's my whole lemon tree. All right. Here are your questions. I did a search. I said baby with a sour face, and that was by far the cutest baby. All right. So think, pair, share. Go ahead and think amongst yourself, and you have 4 choices there. I'm seeing this answer popping up. All right, count of 3, hold it up. 1, 2, 3. Okay. If I could have remembered who the biochemistry students were, that would be a good one. All right, so who wants to tell me why they picked what they picked? Get a bag of candy. Here we go, right there.
I picked what I picked A because it's processed candy, and I didn't think it would need any like actual fruit to make.
Okay, all right. Before we give them the prize, let's have a competition. Who wants to give an answer that might give me a little more biochemistry? All right? You can't, Eric can't do it again. Up there, go ahead.
We'll spread this around.
You're Eric, right? You got a bottle of cider already, so we'll go to the next one. The students in my class, they love to raise their hands. You know why? Because about 2 months later, they're going to come and ask for a letter to medical school. And they said, don't you remember? I always raise my hand. All right, why did you pick, you picked A, it looks like. Why did you pick A?
I thought there was a receptor on the tongue that would register as sour, and they just had to put that chemical.
Okay, what chemical is it?
I have no idea.
No idea. Okay, all right, so, we got another one.
Okay, they probably don't have any lemons. I did say A.
Okay.
And it's probably acetic acid.
Okay, all right, very close. She gets the candy. It's actually citric acid. Give her a hand. All right, so then the question is, what did we learn? So what we learned is that lemons are full of citric acid, but you can make citric acid. That's kind of what we're going to learn. And the second thing is that I thought somebody was going to say, so you guys haven't been students for a while. Lots of times students will tell me, B, C, and D didn't sound right to me, so I picked A. So they had no reason to pick A. Yeah, so, and then D, so the question is, why do I include things like you can't chew beer or can't drink bread? Why do I say it has battery acid? Because the students who are 80% pre-meds, they always say, oh, Dr. Minslow, you're so tricky. You ask tricky questions. So I always try to give them one thing that if they actually picked D, then they're completely lost. And so that way they figure they only have 3 to pick from, a little bit easier. Okay, it does not require any lemons. It has to do with, well, you kind of were both right, because actually you can make citric acid commercially. That was easy. All right. If it does not require lemons, then what does it require to be so sour? Okay, so now we're back to my biochemistry class. We have to learn a little bit here. On the left-hand side is, again, this is from chapter 8 in the book. On the left-hand side, you see lemons. Lemons have citric acid. That's true. However, Aspergillus niger, which is a fungus, it's not Saccharomyces cerevisiae, it's a different fungus, it's a workhorse, it's the white mouse, it's the Drosophila fruit fly of the fermentation world. So Aspergillus niger can make lots of things. It grows in large vats. It actually was harnessed by the Pfizer pharmaceutical company around the turn of the 20th century. In the early 1900s, humans like sour, and confectioner shops were opening across New York City, in the Midwest, certainly in Europe. And what the confectioner, the owners found out is that humans, they like sweet, but sour candy sold better than super sweet candy. And so they were trying to come up with lemon drops and all kinds of ways to sell candy. And so what happened was the lemons were becoming lemony. There wasn't enough lemon concentrate around to do it. And so a chemist in Brooklyn said, we don't need lemons. Let's just go use the citrate cycle and make citric acid. So he did, and he patented it, and it really got the pharmaceutical company going. Why does it work? Because if you inhibit, you probably learned it as a citric acid cycle or the Krebs cycle or the TCA cycle. It's not. It's a citrate cycle. And so when I put the book out there, I had reviewers go, why are you calling it citrate? I said, what's the pKa of the carboxyl group? 4.0, right? What's your pH in your body? 7, right? So you're 3 pH units above the pKa, it's going to be deprotonated. Any carboxyl group that's deprotonated is not an acid. It's a citrate, not citric acid. So I taught the students it's a citrate cycle. I don't call it the Krebs cycle because I like Edwin Krebs. He was a great guy. I never met him, but he obviously helped us out. But that doesn't tell you what the cycle does. It just honors Edwin Krebs, all right? Tricarboxylic acid cycle and citric acid cycle, it's not an acid cycle inside our body, so it's citrate. On the right-hand side, what you see on the top is I got my pointer here. This is the citrate in the media that this Aspergillus is growing. And so what they do is they take sucrose, and at Pfizer, they took sugar beet, which was super cheap. They put sugar beet into the ferment, and the sucrose was being converted to citrate over time, and the biomass stayed about the same. So right here, for any of you that were in production, that's the sweet spot because the biomass had stayed the same, but the Aspergillus were simply converting the carbon in sucrose to citrate. So that was a win-win. And then down here, here's all the things. Some of you may have worked in the medical field. You add citrate so the blood doesn't coagulate. It inhibits the coagulation cascade. Certainly if you have a dishwasher, you're probably putting some citric acid actually in there and some of these sodas. Okay, and then the way we have to do it in my course, if you're going to take an exam on this, this is actually the pathway. So the glucose from the sugar beet, it comes in, the citrate cycle is inhibited because there's so much carbon, plus the Aspergillus has a defect in citrate lyase. So the carbon comes in here, goes to citrate. Citrate is pumped out of the mitochondria, goes out, and it's secreted into the media. They just harvest it, and they sell it as big bags, and to your tongue, basically what he said, you can't tell the difference if it came from a lemon or not. Now, the lemon essence gives your brain more lemony kind of sense, but the pure citric acid, the pure what's hitting your tongue there, that's a brain sour, and that's a Aspergillus. Okay, one more. I think we have time for this last one. All right, everybody ready for this one? This is the one that appeals to you. I picked it specifically for my audience. Why does broccoli, cauliflower, cabbage, beans, why does broccoli cause intestinal gas? Okay. Your 4 choices. This is a figure from the book we had. So this is that's chapter 13 in the book, and I had this story which we're going to tell you. And the publisher, the editor, she says, says, Roger, I don't understand. Like, how are you going to show that the person has a stomach ache? And so I said, well, there's all this, stuff going on. And so they finally came up with this one. And there's little gas molecules coming out. All right, so let's have you think, pair, share. All right, do it twice. Go ahead and think about it and see if you can pick the right answer.
Can you give me the prize?
Okay, this one killed me. You guys might know this. My wife takes Beano. I don't take Beano. I went to CVS last night to get this, and I go to check out at CVS. This was $18. I'm like, are you kidding me? And the guy goes, no, that's what Beano costs. So that's a real prize there if somebody likes it. I think I know why it's so expensive, and we'll actually talk about that. All right, 1, 2, 3. Put up your answer. Perfect. All right, who wants to explain why they picked it? Why they picked the answer they picked? $18 of Beano. You could sell it to your neighbor even if you don't use it. Raise your hand high if you want to answer this one. Nobody wanted to raise their hand because they don't want their friends to know they take Beano.
It's an anaerobic reaction.
Okay, so let's keep the microphone. So why is it that if you don't eat those vegetables, you don't get gas? When you eat the vegetables, you get gas. So what's the difference between eating, say, a piece of pumpkin pie or eating a broccoli? Why would that happen?
Maybe it's high in cellulose.
Okay, there's something about the broccoli or something about it. Do you think the bacteria in your stomach are the same when you eat pumpkin pie or when you eat the broccoli? Yes. Okay, so there's something about the broccoli that the bacteria like that's different than the pumpkin pie, right? Okay, give her the Beano. She's pretty close, darn close there. They can split the bottle because they worked as a team there.
10 for you.
Okay, so anaerobic was a little bit far off, but you did talk about bacteria. All right, so the answer is bacteria convert plant carbs, carbohydrates, to methane. Now, what I didn't say is which carbohydrate. Like, because cellulose is a carbohydrate, so is glucose, so is sucrose, so is fructose. So what carbohydrate? And is there a carbohydrate that's in broccoli that's not in, say, a piece of pumpkin pie? All right, so last biochemistry lesson here. On the right-hand side, these are special oligosaccharides. They're called the raffinose series, and they're 3, 4, and 5. So 3, sucrose, galactose and a sucrose, 2 galactose and a sucrose. That's stachyose, raffinose, and verbicose. 1, 2, 3 galactose and a sucrose. All right, so in these vegetables, they have a lot of this oligosaccharide. So raffinose, the plant itself stores its energy, of course, has cellulose and has starch, but it's actually builds up these 3 different oligosaccharides. All right, so a monosaccharide is glucose, disaccharide sucrose, oligosaccharide 3, 4, 5, and then you go to polysaccharide, which would be starch or cellulose. So these are oligosaccharides, and these vegetables have them. Why do you get gas? All right, so it turns out that the bond that needs to be cleaved in order to harness, to metabolize, is an alpha-1,6 glycosidic bond. Alpha-1,6. All right, there's my 6, there's my 1. That's the carbon 1 on that galactose. There's an oxygen, and then that's the 6th carbon on the galactose below. So it's an alpha-1,6. All right, so that would take an enzyme called alpha-galactosidase. We don't have it. Okay, so now you eat broccoli, goes through your body. You don't have alpha-galactosidase. You can't break the bond. So the bacteria are happy. They're like, yippee, we get it. That's now for us because it wasn't something like amylase, like starch, which you can digest. And so the bacteria that are in your microbiome, they have the enzyme. They digest it, and when they digest it, they ferment it. And the product that they make is methane gas. And so what's happening is the microbiome is these bacteria competing with your own enzymes. You don't have the enzyme, so they win. The product of their reaction is methane gas. So all of a sudden you're feeling all kinds of gassy, all right? It's methane. Methane gas, if you ever did the thing where you, we'll just do flatulence and you light a match, that's it, it's methane. All right, second part of the question. How does Beano work? Okay, so what is Beano? All right, so here are your choices. Hopefully do my Thinkpairshare. All right, here's your last question. So now, and actually, we got one more. I had one candy left over, so we're gonna double up there. All right, so here's your question. How does Beano work? Okay, 1, 2, 3. All right, you got that. My lessons are working. The class is doing well. Okay. Who wants to explain why they picked whatever they picked? Raise your hand. You're gonna get the. We got one right here. You're gonna have to truck all the way over there.
Look at you.
Okay, fine. Wait, let's. Nicole, this person's gonna explain for us back here. And if her answer is because it's on the box, that we're not gonna go with that. Most people in their eyes aren't good enough. I certainly can't see that. All right, I want you to explain. Somebody back here, you raise your hand right here.
Can you raise it again?
I'd say you have a sour tooth because you're actually getting sour candy. All right, so why'd you pick D?
Well, I believe it's D because it would metabolize it higher in the gut. And so the bacteria lower would not have the opportunity to form methane. But the one thing with that is the stomach enzymes usually will break down proteins, and enzymes are mostly proteins. So I'm not sure about that.
Okay, very good. But before you give up, what have you spent your life doing? What is your background?
I'm a physician and I was a chemistry major.
Okay, so fine. I had a feeling. All right, so she said two things. One is she said she wins, so she gets that one. Well, she said it's the enzyme. It is. The Beano is there's enzyme in there, okay? The second thing she said is she knows, as I do, a lot of drugs that might work, they can't survive the stomach, and they can't survive the liver either. So they just don't work. So a pharmacologist's job is to figure out why a compound that works perfectly in the lab doesn't work in the clinic. That's because the stomach digests it or whatever it is. So it turns out that it works, though. All right, so if anybody takes lactase pills, it's all about timing. You got lactose intolerance, take the lactase pill. This is all about timing. You got to get the enzyme in there quick enough to start to digest, and it doesn't always work. But if it works, if you take it, once you have gas, it's too late. You have to take it when you're eating the broccoli. So you have to love broccoli, love cauliflower. Take your Beano. Okay, so then that's how it wins. All right, so very good. So it contains active alpha galactosidase enzyme. Why is it $18? That's an enzyme. And where would you get that much enzyme? How expensive is that to get? You can't synthesize it. It's not a compound you make in the lab. You actually have to purify it. Okay, so where is it? Now we're back to my last biochemistry slide. Here's Beano. It's actually Aspergillus niger again. So now the workhorse is back on duty, and they figured out a way to get the Aspergillus niger. It's just a different view of the same fungus. And it actually will secrete alpha-galactosidase into the media. And they harvest it, they purify it, they put it into a pill. So I think the processing of getting that much enzyme that's still active, not inactivated, is probably not cheap. So there could be a shortage of this, and so that's why it's so high. This is where that other picture came from, because when I showed the editor, she says, well, what do you show in your class? And I go, well, I show this little baby with a big old methane in there. So then they went with something else. All right, so the last little story there. So so the company that came up with this, did they try to solve everybody's problem in the room? They could care less about you. They just tell you don't eat broccoli. What happened was it came from the hog farmers. So the hog farmers were switching what food they were feeding to the hogs down in Iowa. And somebody noticed that when they switched food, which was leftover garbage food that had a lot of raffinose vegetables in it, the hogs stopped eating. And they weren't eating nearly as much as they did before. And so they weren't putting on weight. And they went and they looked at the cheapo food they were feeding them, and they noticed that when they tried to eat that food, they stopped eating. So what they figured out, these hogs had gas, their stomach hurt, and so they didn't want to eat anymore. And so if you're a business person, you'd say, well, I'm not going to feed them hog chow. That's too expensive. I have to go to Purina and get hog chow. The pork will cost too much. I want to feed them the cheap food. So how do I feed them the cheap food? I got to figure out why their stomach hurts. So then that's how they discovered it was the enzyme. So then for 10 or 15 years, they were generating large amounts of alpha galactosidase, selling it to the farmer. Then some bright kid who went to college told his dad, hey, we could sell that stuff because remember your mom has a stomach ache. So they converted what was the agricultural product into a commercial product, sell it at CVS. So that's a good story. Okay, so here we go. Last slide, biochemistry is fun. Didn't you have fun tonight? Of course you did. Everyone's smiling. Biochemistry fun. I'll end with that and take questions. Okay, you guys have the microphone.
So one of the questions is, why doesn't everybody need Beano?
Why doesn't everyone need Beano?
So if it's a particular enzyme that humans don't normally have that then causes you to end up having gas, why do some people have that and some people don't?
Okay, that's what I thought you meant. Okay, so there's, the answer today is much better than the answer was 20 years ago. Everyone's microbiome, which is what bacteria colonizing you have, it's likely that somebody has a slightly different colony group in there. And so if someone reacts very vehemently, they have a real problem with broccoli, my guess is they have a lot of the bacteria colony in their microbiome that really works really well and converts it very quickly. And somebody else may not have very many of those bacteria. So it kind of goes back to the one we said before. If you have the right kind of biome, you might not have a problem. Okay, question? Mm-hmm.
My question is, we're told that cattle produce copious amounts of gas. Why hasn't someone figured out a way to get this enzyme into cow chow?
Yeah, so that's a good question. So I think that it's slightly different, first of all. So the gut, the second stomach, and I'm sure there's ranchers in here somewhere. The second gut, of course, has that other group of bacteria that help them digest cellulose, right? So cows, they're not so much eating these fancy vegetables that they need to eat. They're eating like hay and grasses. So a cow is asking the bacteria to actually break down cellulose, okay, just a pure plant cell wall, and there's enough sugar in there for them to take advantage of that. So it's slightly different, but yeah, methane gas from cows contributes to climate change through global warming, no doubt about it. I think the reason that's an issue, in fact, anybody, if you've seen any of the Food Inc. or any of the documentaries, if the way to get that protein, which is what a cow is, just a walking thing of protein, but you're feeding it cheaply enough so that the beef doesn't cost too much, right? Because if you switch to a more expensive food, then it's going to cost more. So that's the trade-off, but it's slightly different than the Beano one. Okay, other questions?
Can you tell me your thoughts on probiotics and how they compare to the probiotics in yogurt?
Yeah.
Is there a difference in should I buy the good stuff?
All right, so I'll answer the second part first. We don't know enough, I don't think, at this point to really know which of the natural products are better or worse than others. There's two reasons for that. One is we're not sure what's in the natural product exactly, because there could be 250 ingredients in there. Second, we don't know enough about what's in your biome to know if the mix of this plus that is going to work. That's the second part. So we don't know what's going to work or not, so I can't tell you what to buy. The first part is about probiotics. So probiotic is often misused. And I actually have it in my book. You can go and buy the book. It says probiotic is pro-bio. Okay, so when a lot of people think probiotic means you're eating bacteria, which you can. If you eat a live culture of yogurt, you are eating live bacteria. Now, it doesn't mean that it actually takes residence in your gut. But a probiotic can also be a compound that stimulates the growth of gut that's already in there. And the one that I use, I'm looking at the physician now, is human breast milk. So in human breast milk, there are over 100 different sugars. And they've isolated 3 of the sugars that they know what they do. And 2 of the sugars that are in human breast milk are probiotic. They're full on, like, what makes them a probiotic? The one sugar, which is a form of tetrose, it stimulates the growth of a bacterial species in the baby's gut that is thought to be healthy for the baby. So when the baby's drinking human breast milk, that sugar is very well metabolized by that one bacterial strain. And so now that bacteria takes off, and that bacteria is protective of pathogenic disease for the baby. Okay, so that it's called probiotic, but obviously the baby already had the bacteria. The second one is a form of a sugar that is a decoy for pathogenic bacteria that would otherwise give the baby intestinal infection. And so what this other sugar does is it looks just like the sugar on the intestinal cell of the baby. And this sugar, which comes from the woman's breast milk, binds to the lectin, which is the receptor on the pathogenic bacteria. And now the bacteria is plugged with this sugar that's just coming through on the breast milk, and it can't bind to the intestinal cell. So that's a probiotic because it's now acting as an antibacterial. So I think the way to think about it is eating live bacteria in kombucha, which my wife drinks, is probably gives you some bacteria, but how much of it actually stays and if it really colonizes, who knows? But you should think more broadly about what a probiotic is. It's a compound that makes changes in your biome. The thing about fecal transplants, probably people have read about that. It seems kind of odd, but it doesn't seem odd if you think about it from the standpoint of what you get. So a fecal transplant is where somebody has a microbiome that's considered the great colonies of all the perfect combination, and their fecal material, of course, carries live bacteria. So some people are using these to now inoculate somebody else. So that's a little bit far-fetched, but what it tells you is a microbiome could be transferred through that, and not everything is thought of as pathogenic. Okay, any other questions?
I know you said you weren't going to talk about mosquitoes.
Yes.
But we seem to have an awful lot of mosquitoes this year. Do you have any idea on why that's happening?
I have the same idea as you have. We have a lot of mosquitoes in Tanque Verde this year, for sure. We certainly had a lot of water. I know, I'm not an entomologist. She's an ecologist. Do you know why? Can you tell us, Nicole? All right. So the population, I know one thing I do know about mosquitoes is that when she lays her egg, it only takes 72 hours for the egg to hatch. And then the larvae grow into pupa, and then they eclose in about 7 or 8 days. So the monsoon rainy season depends on how long of a dry is between the rain. So when it comes frequently, she can lay a lot more eggs if there's humans around or animals to bite and get the blood to do it. So this year, it could have been that the population got large enough, the mosquito population, that as it rained the next time, it just went bigger. Okay, so if you had dry times, it could be as many 10 days or 15 days with no rain in August, when normally it rains almost every day. Then you could break the cycle, and then the population would go down a little bit. The other thing that makes it terrible for humans is Aedes aegypti, the one that we study, it bites humans 98% of the time. And the reason it prefers humans, therefore it colonizes flower pots, anything around a pool, colonizes anything there's a divot, and so they tend to invade urban areas, and then that becomes more of a pest, actually.
I'm sorry.
All right, good.
if you empty them as soon as it quits, you eliminate probably 80% of the breeding area that these mosquitoes have. You can't eliminate divots in the ground unless you want to go out and fill them. But if you just do this little bit of housekeeping, I think you could eliminate a lot of mosquitoes around the house.
Absolutely. And I think that's a good point. So two kind of comments since you brought up mosquitoes. Malaria, which is a much more pathogenic, it kills many more people a year, about 2 million a year, more than dengue. It's really hard to control anopheles in Africa because when you think you're in a jungle and you think of not flower pots, you think of every divot, every little rock, every little thing. And so the reason larvicides, we think of putting those mosquito pellets in like flower pots or wherever there's something because you can get there. But the real killer, the real plasmodium, the real killer is these anopheles mosquitoes, which you can't possibly do it. So you have to kill the adult. So the way you kill the adult mosquito is you put it in the bed nets because now you're sleeping in Africa underneath the bed net and the mosquito comes to the net. So in where we live, Aedes aegypti, you can certainly make a difference by simply knowing where all the water spots are, and as soon as it rains, empties them out, and that makes a big difference. The second story is my sister-in-law is Austrian, hardcore Austrian, lives in the United States. She's got a million rules, a million things that happened, everything she learned in Vienna. And so I study mosquitoes. I'm at her house in San Diego, and she's got this bucket, 5-gallon bucket, with water in it. And I look in there and I'm like, Rebecca, you got larvae in there. She goes, yeah, I know. I go, why do you do that? She goes, I time it. I fill the bucket and then I mark it on my calendar and I check it every day. And as soon as I have enough larvae in there, it acts as a trap for the female to pick that spot. And then she pours bleach in it, pours it out, does it again. So what she's thinking in her head, which must work a little bit, is she's essentially setting up mosquito traps, but she's very careful to watch them. And I just said, well, that's kind of crazy because if you're not watching them, you're now breeding mosquitoes. But she said, you know, where people live, some places in Europe, some of these swear by it. As long as you keep the trap and you empty it, it's a pretty inexpensive trap. So I wouldn't recommend that, but that would kind of make sense that you actually bleach it before they pupate.
I knew he was going to ask. Yeah, I have a mosquito complaint too. The ones I see in our yard are smaller than I'm used to seeing, and they're coal black. It looks like they're tinier, and they're I call them stealth mosquitoes because they sneak up on you on the backside and bite you on the leg someplace where you're not looking, or on the elbow, or the knuckles while you're busy doing something else.
Yeah. So okay. So the ankle biters are Aedes aegypti. And they can be tiny if they're not healthy. The ones we grow in the lab are pretty big. The other one we have here is Culex pipiens. And it's actually called Culex pipiens. They're brown, kind of light brown. They're really tiny. They carry West Nile virus when they're infected. So the two main ones are Aedes aegypti, Aedes albopictus, which is a relative, but it still carries dengue and Zika, and then Culex pipiens. So the size range is different, but if it's blackish, and if you looked under a microscope and you saw white lines, that's Aedes aegypti, and they come out in the morning and at night, and they prefer ankles, and they're really annoying.
They prefer my ankles, and my wife doesn't get blood with them at all.
Yeah, they had yeah.
In fact, as soon as I walk out the door, they zero in on me. I mean,
you know, that's the question that some people say, go back to like why some people need broccoli, some can't. Why are some people bit by mosquitoes more frequently than others? And there's tons of studies on that. So your sweat, your smell, some is more attractive to mosquitoes than others. My guess it's not evolution to be the one who feeds the mosquitoes. It's evolution not to be the one that feeds the mosquitoes. So that sort of happens. The second thing related to that is in Africa, they made a trap, but not the trap with the water in it. They made a trap with dirty socks. You might have heard about this. So Limburger cheese has a smell to it, and it turns out it's a fungus that infects your feet and makes your socks really smelly. That's a fungus. That's a smell of a fungus. And it's the same version that's in cheese. And so what they figure, everyone in the village put their dirty socks in this big thing. And it actually worked. And then they would empty it out at night. And they finally traced it back to the mosquito is smelling fungus. And a lot of people have it especially in jungles, on their feet. So that's how it zeroes in on the human foot, and that's why it stings there. I mean, bites.
Are these little black ones that I'm talking about, are they the Egypti?
Aedes aegypti, be my guess.
And so we ought to worry about those carrying some kind of.
Okay, so good question. So dengue has been, dengue is the virus. There are Aedes aegypti mosquitoes infected with dengue in Brownsville, Texas, in Miami, and in Galveston. If you go 200 miles south into Mexico or go to Dominican Republic, there is a much higher prevalence. In fact, Dominican and Haiti have a lot of dengue there. So what they think is happening is the mosquitoes certainly live here. And so the question is, why isn't the pathogen infecting all of the Aedes aegypti here in Tucson compared to the Aedes aegypti 500 miles south? And there's two reasons for that. One is it's drier here, so the mosquito is not as healthy. But the second one is this relationship between host pathogen and vector. So if a lot of people in the village in Mexico have dengue, then when the mosquito hatches, she gets dengue from the next person she bites. But if only 1 out of 10,000 people up here, even though the mosquito could technically carry it, doesn't carry the virus, the mosquito's not born with the virus. The virus doesn't go from mother to egg. And so the newborn mosquito has to bite an infected person. So you can imagine how many PhD theses were centered on a model in which you say 20% of my mosquito population during the rainy season has dengue virus. 10% of the human population in the village has dengue virus. What's the probability that I'm going to get the mosquito population up to 40% and my human population up to 50%? So you can model the whole thing. And what happens is the reason why we want to decrease mosquito populations during the rainy season in high transmission areas is because you get below the threshold of the model. And then you have a dramatic effect on whether you're going to get new infections or not. But if you don't empty the pots and nobody's paying attention and nobody's going to the doctor and they're infected, then the whole thing it's a mess.
I have one final question. We live at 3,300 feet above sea level, and these mosquitoes have no trouble flying around, but they're so tiny. What's the altitude limit of a mosquito?
Yeah. Well, I don't have all the answers to the mosquitoes, but I do know that at my house, we turn the fans on a lot, just because the airflow is one way to kind of keep them from landing on you. It's not a bad idea. Citronella are all the things you can put in those little traps and stuff. I think they kind of keep it down, but I go back to his thing. Just don't have standing water in your house if you.
Well, I'm just wondering what's the altitude limit that these insects can support themselves in flight. In other words, how high can a mosquito fly?
Yeah, I don't know. I don't know much about that.
Well,
all right.
At least
any other questions? One back there. It's almost like, it's sort of like, I shouldn't even told you I worked on mosquitoes.
This is going to be non-mosquito.
Okay.
In upcoming lectures, are you going to have anything on the applications of biochemistry to cancer treatment and other things like that?
So one of the lecturers is Dr. Wolfgang Petty. He is a biochemist with a degree in chemistry, and he is studying a protein called phosphatases. So kinases are enzymes that phosphorylate, and there's a lot of kinase inhibitor drugs that treat cancer. His whole research program is saying that if you can activate a phosphatase, it's the same thing as inhibiting a kinase. So a kinase puts a phosphate on a group, and that phosphorylation could be cancer driving. So you inhibit the kinase so that it's not phosphorylated. The cancer protein then is inactive. So he's trying to activate phosphatases, which remove phosphates. It's very fascinating stuff. It's tricky because the drug has to fit between two proteins. The kinase is great because it's an enzyme, and you can put an inhibitor right inside the pocket of that enzyme. That's why most drugs inhibit enzymes. The phosphatase regulators he has to inhibit are ones that are at the interface, so he has to find molecules that fit, and he'll show you some beautiful pictures on that.
All right.
Any other questions? What another?
One last one.
Last one, real loud.
Last one. Come in.
Can you break it down? down, or how we break it down?
Yeah. So, that's great. So, when you drink your libations, your liver has enzymes that are going to metabolize the ethanol. And so the drunk feeling that you get is the level of ethanol in your blood, but of course it's being broken down by your liver. And so so that's how you know it's broken down, because your blood alcohol levels go down over time. And as the blood goes through your liver, which happens every so much time, the enzymes in your liver are going to degrade the ethanol. Yes, it is. It is. Because the reaction can go back. That reaction can go back. So you can get acetaldehyde by converting ethanol back to acetaldehyde. And there's enzymes in your liver that can go backwards. So basically what it's doing, the reason why you get that sort of drunk, fuzzy feeling, of course, is in your brain, the ethanol is affecting neurons in your brain. So as your ethanol levels drop in your blood because it's being modified, metabolized by the liver, then of course you stop interfering with those pathways. And if you eat a really, what's a hangover meal, or what you do if you don't want to drive if you haven't eaten for a long time and had too many bottles of my cider, because now your ethanol levels are high. If you eat a fatty meal, like a Sonoran hot dog or something, that fat, because the ethanol will be absorbed by what's in your stomach, then you can absorb some of that, and then it's not in your blood. All right, thank you very much.
Thank you. Sorry for coming. I just wanted to announce what the next talk is going to be. The next talk is Thursday, November 8th, 6:30 p.m. And it will be Matt Cordes, and you will present Venoms, the Chemistry of Life and Death. And we hope to see you there. Thank you.